Hi everyone, my name is Brittany Robertson and I'm a current PhD candidate at Murdoch University. I recently completed my internship at the Pawsey Supercomputing Centre investigating gene targets for crop genetic improvement. Starting off with a bit of background about me. I completed my undergraduate degree in molecular biology, which led me to study a PhD in plant functional genomics. For my PhD, I'm investigating target genes for improving crop drought tolerance using the crop barley as a genetic model. Stomata are an excellent candidate for improving drought tolerance. So what are they? Stomata are essentially tiny living pores on the leaf surface. We can see in this video that guard cells surround the central pore and can adjust its size or aperture. Stomata serve as an entry point for carbon dioxide to enter for plant photosynthesis, but are also a point of exit for water to escape. In fact, over 90% of plant water loss occurs through stomata, so they are a major target for the improvement of crop drought tolerance. Stomata are present in almost all species of plant. Despite this, plants have adapted different stomatal designs and functions over time. For example, grasses like barley and corn have dumbbell-shaped guard cells paired with subsidiary cells on each side, which allow for better fine-tuning of the pore in response to environmental signals. On the other hand, eudicots like Arabidopsis have more simple stomata that consist only of the guard cell pair. These differences are because of divergences in key genes for stomatal development. In this central figure, we can see that multiple genes have diverged in function for stomatal development in the grass family versus Arabidopsis. Through comparing the genetic material of different varieties of the same crop species, we could find a mutation in a gene for stomatal development possessed by a variety with high drought tolerance that may be used for future genetic improvement. This project aimed to identify novel genetic variants or polymorphisms for genes controlling stomatal development through investigating the barley pangenome, but we have some problems. The barley genome in itself is huge at 5,100 million base pairs, but we aren't just looking at one genome, we're investigating the pangenome, which consists of all currently sequenced barley varieties. All in all, this means we need to process over 387 billion base pairs of data. Such a large amount of genomic data requires much greater computing power than just a home PC. Otherwise, it would take forever. This is where Pawsey systems come in. For my project, I used both the Cytonix and Nimbus systems. Cytonix is an incredibly powerful supercomputer. Cytonix was very useful for performing tasks that required large amounts of RAM, for example running genome projections in my project. I also made use of a Nimbus allocation. Nimbus was flexible as it allowed me to build my own environment from scratch and enabled the completion of smaller jobs on my own Slurm managed cluster. Following my initial phases of analysis on Pawsey systems, one trend was immediately apparent. There was very large variability across the barley pangenome for genes of stomatal development. This pangenome graph represents the variability for all the stomatal genes across all varieties. Notice the large amount of edges or lines that curve into the graph over and over again. Edges represent points of genetic divergence across the barley pangenome. In this project, high levels of variability were discovered for genes of stomatal development across the barley pangenome. The next step is to reperform alignments with smaller flanking regions to gain enhanced clarity of the nature and location of sequence divergences between varieties. Novel polymorphisms were also identified between select barley varieties for genes of stomatal development. Here our next step is to identify genes possessing polymorphic regions of high significance, and to investigate the effects of these novel polymorphisms on drought tolerance through bioinformatics and functional characterization experiments. I would like to thank my supervisors, Pawsey mentors and staff of the Pawsey Supercomputing Centre for their excellent support during my time as an intern. This internship has provided me a solid foundation to build my computing skills in preparation for a career in genomics.